will present slides about GPCR or G protein coupled receptor. So we all know about cell surface receptors. Three types of cell surface receptors are present. One is ion channel linked receptor. The second one is G protein linked receptors. And the third one is enzyme linked receptors. So in G protein linked receptors, two pathways should be followed. One is cAMP or cyclic AMP pathway, and the another one is IP3 DAG pathway. Here I will discuss about GPCR or G protein coupled receptor and the cAMP pathway or cyclic AMP pathway. So in the picture that is shown in this slide, we can clearly see the G protein coupled receptors in inactive state and in active state. In inactive state, simply GTP is binded, and in active state GTP is binded. So before knowing all about this, we have to know about G protein coupled receptor. This G protein coupled receptor, we clearly see it is 7 transmembrane domain and or serpentine receptors. This is a serpentine receptor, and here 7 transmembrane domains are present, and here 3 subunits are also linked. One is alpha, another one is beta, and another one is gamma. It is present in heterotrimeric form and binded with each other with the presence of GTP in inactive state. And here no ligand is present, so it is not activated. But when ligand is present and binds with the receptor of this serpentine domain, then what happens? Then GTP converts into GTP by the action of GEF or guanine nucleotide exchange factor. Then this GTP binds with the alpha subunit and breaks the alpha beta-gama. Beta-gama separated from this alpha subunit in dimeric form, and alpha is in monomeric form, binds with the GTP and activated. These activated G protein coupled receptors now activate another proteins or enzymes and do further cell responses. So it is simply, and we all know that GDP is converted into GTP by the action of this guanine nucleotide exchange factor or GEF, and also GTP is also can be converted into GDP by the action of GTPase. This GTPase is a largest group of enzymes, and this GTPase is activated also by RGase or regulatory of G protein signaling. Now simply, when GTP is binded with the alpha subunit, the trimeric form breaks down, beta gamma releases from the alpha subunit or separates from the alpha subunit in dimeric form, and alpha is in monomeric form, do further cell responses in active states. And when GTP is binded with the alpha and there is no ligand in the cell surface receptor, then alpha beta gamma is present in heterotrimeric form, and the serpentine receptor G protein coupled receptor is not activated and don't be able to do further cell responses. So for the cell response, we should have to convert GTP into GTP. After that, what happens? Now for the next one, the cyclic AMP or cAMP pathway. In this cAMP pathway or cyclic AMP pathway, what happens? This GPCR is present in the cell surface and ligand is binded in the receptor. Then alpha, beta, gamma. These subunits are separated when GTP is binded with the alpha subunit. And this activated alpha subunit then activates adenylate cyclase. This adenylate cyclase or AC converts AMP into cyclic AMP, and also cyclic AMP can be converted into AMP by the action of PDE or phosphodiesterase. So when AMP converted into cyclic AMP by the action of adenylate cyclase, more and more cAMP molecules are present or produced. Then what happens? We all know about pKa or protein kinase A enzyme. This pKa is cAMP loving enzyme, and in inactive state there is two regulatory subunits and two catalytic subunits binds with each other, and these regulatory subunits or each regulatory subunit have two receptors, cAMP receptors present in it. So when these cAMP molecules are connected with the regulatory subunits or receptors of the regulatory subunits, then what happens? Then these regulatory subunits separates from the catalytic subunits and separates from each other. Then these catalytic subunits enters into the nucleus from the cytosol, and there is a CRAB, or this CRAB is cAMP response element binding protein present in the nucleus. Then this catalytic subunit activates the CRAB, and this CRAB is become activated and stimulates the gene, and then it does transcription, and then translation and further cell responses happens. So this is the short summary of cAMP or cyclic AMP, how it happens. This cAMP is also called second messenger in this GPCR. We should have to be memorize this. After that, there is a short summary about this. So when no ligand is present, GTP is binded with the alpha, beta, gamma trimeric subunit. In inactive state, then ligand comes, ligand binds with the serpentine receptor, and then alpha and GTP binds with the alpha subunit and breaks beta gamma in dimer form. And this activated alpha then activates adenylate cyclase, which is present in the cell surface, and then this adenylate cyclase converts ATP or AMP into cyclic AMP, and more cyclic AMP is produced. Then protein kinase A or pKa has two regulatory subunits and two catalytic subunits. Each regulatory subunits have two receptors or cAMP receptors, and then this cAMP receptors binds in the regulatory subunits and breaks the regulatory subunits from the catalytic subunits. These catalytic subunits then enters into the nucleus and activates the CRAB, and then transcription, translation, and further cell responses happens. Hope you like it. This GPCR or G protein coupled receptors.